Natural selection is when the environment exerts a pressure on a population so that only some phenotypes survive and reproduce successfully. These organisms exhibit Darwinian fitness. Gene flow makes two different populations more similar to each other and can occur through the movement of individual gametes. Mutations occur whenever a mistake in the DNA occurs in the heritable cells of an organism. Genetic drift occurs when the alleles that make it into the next generation in a population are a random sample of the alleles in a population in the current generation. Two examples of speciation are the founders and bottleneck effect. Both have random allele changes and decreased genetic variation in the population. The founders effect establishes a subpopulation as a new colony drawn from a larger population. The bottleneck effect is a subpopulation caused when a natural disaster reduces the size of an original larger population.
The photic zone is a surface layer of the ocean that receives sunlight and is filled with photosynthetic organisms such as phytoplankton and plants. The aphotic zone is the deeper portion of a body of water, and it receives little or no sunlight and is usually deeper than 1,800 meters. The deepest zone of the ocean is the benthic zone. This zone receives little or no sunlight and is filled with deep sea fauna such as tube worms, echinoderms, cucumbers, crustaceans, and corals. These organisms typically acquire their energy from nutrients in the sediment that originates from dead organic matter that has sunken to the ocean floor from the higher levels of the water column. An example of this is whales. Another aspect of the deep sea environment are hydrothermal vents. Vents are essentially convection systems in which water that has percolated through a fissure in the Earth's crust is heated by magma and reemerges through a vent's fissures. Because vents are temporary and appear and disappear spontaneously, there's a resulting bottleneck effect on the populations that live in these areas. These vent populations frequently experience the founder effect. The populations that inhabit this area typically have a free-floating larval stage, which, paired with the ocean currents, greatly influences the dispersal rate of these populations. Scientists recently have been investigating gene flow in the deep sea between hydrothermal vent species.
When looking at deep sea gene flow, it is important to look at dispersal patterns. Deep sea organisms such as mollusks have two larval shells known as protoconch 1 and protoconch 2. Organisms that have smaller eggs and active feeding larvae tend to have longer distance dispersal patterns as they can easily ride ocean currents to new hydrothermal vents. Organisms that travel farther will have gene flow regardless of distance, while those that cannot travel far will have high gene flow at closer distances but lower gene flow as distance increases. The island model can be used to show gene flow for species with small eggs and active feeding larvae. This is because the organisms that possess these qualities have long-distance dispersal capabilities. For the island model, the rate of gene flow between colonies of organisms is independent of geographic distance. The stepping stone model can be used to show gene flow for species with large eggs and non-active feeding larvae. This is because the organisms that possess these qualities have limited dispersal capabilities. For the stepping stone model, the rate of gene flow between colonies declines with increasing geographic distance.
Biological evolution is change in heritable characteristics of a population over succeeding generations. Mechanisms for biological evolution include natural selection, gene flow, mutations, and genetic drift. Hydrothermal vents are fissures on the planet's surface from which geothermally heated water issues. Vents are temporary and spontaneous, resulting in the bottleneck effect. Ocean current generated from vents greatly influences the dispersal rate of the population. Dispersal is the movement of individuals from birth site to breeding site, and its potential is characterized by how far dispersal ability reaches. An increased ability correlates to a greater potential for gene flow. Weak dispersal ability increases distance but decreases potential, and for strong dispersal, distance is irrelevant.